All right, welcome to the first screencast for Honors Chemistry. This unit is the Matter, Change, and Energy unit. This is Honors Chem Unit 1. Some advice to you while you're listening to me and going through and taking these notes, I would have this PowerPoint opened up so that way you can flip back and forth between the tabs. So if you have the PowerPoint from Edmodo opened up, you're able to, go to the actual slides and click on any of the links that are in the slides. Although for this first screencast, we will be doing all the links in class, so you won't have to. You can always go back and click on them afterwards. Remember, you can pause and go back and re-listen to me explain something if you don't think I explain it very well, or if I move too fast. You can pause to slow down so you can catch up on your notes. But I'm going to go ahead and start the presentation. And this is, like I said, the Matter, Energy, and Change unit. We're only going to go through objectives 1 and 2 on this particular screencast. Okay, so objective number 1, identify steps in scientific method and distinguish between a hypothesis and a theory. So this should be a review for you, but we're going to cover this material. So this is what will be included in objective number 1. So the scientific method versus the engineering method, it's a little bit different. In the scientific method, we ask a question. Why does this happen? How does this happen? You do background research to try to figure it out or see what we know. Construct a hypothesis, test your experiment. Is it working? If it's not working, you go back and start over again. If it is working, you keep going, analyze your data, and draw conclusions. Do your results line up with your hypothesis? Do they not? You might have to go back and restart over again, or go and you learn something totally new. And so you go back and construct a new hypothesis, and that's where you get this part of it, where you get new and future projects or new and future research. And you might end up that you were completely wrong, and so you would communicate those results that, hey, this is not what's causing it, or this is not what's producing that effect. The engineering method is a little bit different. It usually starts with a problem rather than a question. The problem would be, how do I get from point A to point B? Or how do we cross this mountain? Or how do we go through a mountain? That's an engineering problem. How do I make this possible? So we do some background research again. Specify your requirements. I need it to get from point A to point B. I need this bridge to span this particular lake. Brainstorm, evaluate, and choose your solution. What's going to be the best possible thing? You would develop a prototype, which is very similar to an experiment, but you're going to make a prototype and see if that prototype works before you make it on a grand scale. You test your solution. You test that prototype. Does it meet what you need? Does it not meet what you need? And you go back and you fix it. So the engineering method and the scientific method are both very similar. But remember, in the scientific method, we're asking a question. And making a hypothesis based on what we think is causing that. Whereas in the engineering method, you are trying to solve a problem and you create a prototype and then you create some sort of a system to solve that problem for you. Okay, so when we're doing an experiment, they're always going to be controlled experiments. These blue links have videos with them. We are going to watch these videos in class, so no need to watch them right now. But a control is used as a standard of comparison. I'm going to give this person this medication and I'm not going to give this other person the medication. That's my control. So I can compare what effect the medication has. The independent variables are what's changed. So the independent variable would be the medicine that I was giving that person and I wasn't giving the other one. Everything else is going to stay the same. That's the dependent variable. So the dependent variables, they change in response to changes in the independent variable, but they're going to be something that I hold constant. So, the people need to be the same age. They need to be, or have roughly the same disease. All of these things need to be controlled for. And those are going to be your dependent variables. And the independent variable is what you would change. A hypothesis is generally going to be written in an if-then statement. If I do this, then this will happen. So that makes it easy to identify, especially if you can take a hypothesis and kind of turn it into an if-then statement. It is an educated guess about what may occur during the investigation. You are making your most informed, educated guess. You're not going to waste your time and perform an experiment on something that you know is not going to work. So, and it's a tentative explanation for observed phenomena. Why is this happening? Well, here's what I think. If this happens, then this happens. A hypothesis, once tested, may become a theory or a law. A theory is an explanation principle. It's based more on inference, but it is based on lots of evidence. Basically, you're going to have a whole bunch of different hypotheses falling under that theory. And it can change with new evidence. I use a lot of biology examples for theories, like cell theory. Everything that we know about cells falls under cell theory. So the eukaryotes, the prokaryotes, every single thing that we know about cells, all the organelles, all their functions, what they do, how the cell lives, how the cell dies, how the cell maintains itself, all of the metabolism, everything the cell does is part of cell theory, this big overreaching kind of concept. A law is something that we generally don't see in biology, but you do see a lot of in chemistry. It's a concise descriptive principle. It is definitely based on more than one observation. It's based on lots of evidence, and it can change with new evidence. But it is not something, we're not trying to explain why it happens like you do with this theory, like evolutionary theory or cell theory or germ theory, that germs and fungus and bacteria and viruses cause disease. So with a law, we think of things like the law of gravity, what goes up and what comes down. That's a concise descriptive principle. There's different laws in chemistry, law of conservation of mass, law of conservation of energy that we will go through and discuss. Okay, so types of observations that we might take when we are doing a scientific experiment or doing a lab. Qualitative would be descriptions using adjectives based upon your senses. So that's going to be with words. Think quality. So yes, thinking quality. Qualitative is going to be word descriptions of things. Examples: color, odor, warm, cold, physical state, if it's a solid, liquid, or a gas. Quantitative is going to be in number form, so measurable with a measuring instrument, so ruler, thermometer, balance. When you think quantitative, think quantity, things that have numbers associated with them. So your actual temperature, how long something is, the mass something has. All right, objective number 2, identify reasons to study chemistry. So what is chemistry? It's the study of matter and the changes that it undergoes. So what the heck is matter? Well, that's anything that takes up space and has mass. Energy is not a form of matter because energy does not take up space. It does not have mass. Energy is the capacity to do work, so you need that energy in order to make things move or make things go or make things happen. But energy is not specifically matter. So a little bit about chemicals in chemistry. A chemical is any type of matter, a substance that has mass and volume. These video clips here, the "Are chemicals really bad for you?" we are going to discuss that in class, as well as do this little brainstorm activity. We will do that in class, so no need to worry about that yet. We are going to watch this video on the 5 branches of chemistry in class, so you don't need to watch it right now, but you might jot down these 5 branches. Organic chemistry covers carbon-based chemicals, so a lot of times you'll see that in biology or biochem. Inorganic chemistry is basically chemicals that do not contain carbon, so that's what we will deal with in here, is primarily inorganic. Analytical chem is where you study the basic composition of matter. Analytical chem people are going to be the ones that are breaking every single thing down into what it's made out of, to the most minute detail, analyzing basic chemicals. Biochem is the chemical processes of living organisms, so what chemicals make life possible. And then physical chem, studying the mechanism, the rate, and the energy transfers when matter changes. A lot of that relates to physics, so that's where physics and chemistry would overlap. This jigsaw activity, so we're going to watch this video in class, this 8-minute video. We are also going to watch this 2-minute video in class, and then we are going to do this jigsaw activity. You can kind of read through what we're going to do just to get an idea of what we're going to do, but we will perform this activity in class, and that is it for objectives 1 and 2.